Hi students, welcome to today's biology class. My name is Dr. Suja Agustin. Today we are going to study about an important topic from your 11th standard syllabus, that is from the photosynthesis chapter, non-cyclic photophosphorylation. Okay. So before going to that topic, first you should understand, right? So we will think about photosynthesis. I'm not going in much depth, just we will understand what is photosynthesis. Whenever you get any word in biology, never try to by heart it, just split and see. So here we can make a split here, right? We are getting photo and we are getting synthesis. So synthesis means formation, photo means light. So it is the formation of something in the presence of light. So already you know it is the formation of carbohydrate by utilizing carbon dioxide and water in the presence of chlorophyll and sunlight. So that is photosynthesis. In photosynthesis we have two reactions, right? One we will call it as light reaction and the other one we will call it as, yes, dark reaction. Light reaction depends on light, right? Whereas dark reaction, it is independent of light. Never think that it is occurring in the absence of light. That's why it is called as dark reaction. No, it's independent of light. So I just want to give you a basic idea. The ultimate source of energy is always sunlight, right? It is always sunlight. In light reaction, sunlight energy is fixed in the form of, this is occurring in thylakoid, okay? So that means grana. It is occurring in grana. Sunlight energy is fixed in the form of ATP and NADPH2. Now, these energy molecules are used in dark reaction to fix carbon dioxide into carbohydrate. Now, when you think about dark reaction, we have C3 reaction, Calvin cycle, C4 cycle, Hatch and Slack pathway, correct? The same way when you think about light reaction, we have cyclic photophosphorylation and non-cyclic photophosphorylation. So, this is the way we are reaching our topic, non-cyclic photophosphorylation. So, let's see the details now. Loads of question can be asked for your NEET exam from this non-cyclic photophosphorylation. So we are going to the topic non-cyclic photophosphorylation. Non-cyclic, right, photophosphorylation. Okay, I will write here. That will be better. Non-cyclic photophosphorylation. Now look at the spelling. Photophospho-rylation I have written, but actually when we pronounce, we will pronounce it as relation only, okay? So non-cyclic photophosphorylation. So now we are going to split the word as such. What we studied in photosynthesis also, right? The same way, whenever you get anything in biology, never try to by heart it, okay? Just break. So first we will look at cyclic, non-cyclic. So you are getting two words here, non and cyclic. What do you mean by a cycle? A cycle means it starts from a point, it ends in the same point. Now when I am telling it is non-cyclic photophosphorylation, you understood that. It starts from a point, but it never come back to the same point. It is not coming back to the same point, right? So that's why it is called as non-cyclic. Now, what is photophosphorylation? Again, split and see. Here, if you make a cut here, you can see photo. Photo means light. Phosphorylation means it is a formation of ATP. From where ATP will be formed? ADP, adenosine diphosphate, right? Plus inorganic phosphate will give you ATP, adenosine triphosphate, right? So ATP is formed, the energy currency is formed in presence of light. That's why we are calling it as photophosphorylation. So what do you mean by photophosphorylation? Photophosphorylation is a method where the energy molecules like ATP is produced in the presence of light. Now why it is called non-cyclic photophosphorylation then? Because this method of ATP formation occurs by the moving of electron in an electron transport chain. So there is a primary source. Just imagine there is a source from where the electrons are emitted. Actually, if it is coming back to the same source, we can call it a cyclic. Here you can imagine it is not coming back to the same source. Instead, it is going to another source. Okay. So non-cyclic photophosphorylation, it's a method where ATP molecules are produced. In that method, the electron which is emitted from the photosystem is not coming back to the same photosystem. Okay. Now, this is the schematic representation of non-cyclic photophosphorylation. This is not an easy topic. Okay. Now, if you look at this representation, you can see a zigzag, or you can see Z shape is also there. So this method is otherwise called as Z scheme. Okay, this is otherwise called as Z scheme. Now there are many molecules, right? This you have to by heart. Okay, the sequence you have to by heart, then only we can. So you just by heart 680, pheophytin, QA, QB, cytochrome B6F complex, plastocyanin, P700, then A0, A1, FeS6. Okay, here there is FeSa, then FeSp, then ferredoxin. Okay, FeS6, FeSa, FeSp, ferredoxin, NADP reductase, NADP, NADPH plus H plus. Okay, and here we have one oxygen evolving complex. So this sequence you have to by heart. Okay, if you want to write an essay, just like a 5-mark question can come for your board exam. If you are using this for that purpose, then you have to by heart the sequence. Okay, it will not take that much time. It will not take that much time. So normally we have to start this procedure from here only. Actually, light energy will fall on P680. Like that we have to start. Then the electron is going to P700. But when I am going to explain this, okay, I am starting from P700. Reason I will tell you later. When you think about this process, no, understanding level, if you study from P700, there is a logic in it, so you will be able to follow in an easiest way. Okay, so what is the ultimate source of energy? Yes, sunlight. So sunlight energy fall on P700. Now what is P700? PS1, photosystem 1. Now P680, this is photosystem 2. What do you mean by photosystem? Photosystem, so it's a light absorbing complex. This is present in the leaf. Photosystem, it possesses chlorophyll molecules, protein molecules and all. It has a reaction center. Okay, this you have to remember always. It has a reaction center. Now here the reaction center. Okay, how to remember? 1 is odd number. 7 is an odd number. For P700 you can connect with PS1. Now PS2. Okay, 2 is even number. So 6 and 8 are even number. So PS1, the reaction center is P700. Odd we are connecting because I know we have only 2 wavelength we have to by heart, 680 and 700. And you know we have PS1 and PS2. During NEET exam time, within 30 seconds you have to find out the answer. Normally the question will come, which is the reaction center for PS1. So you will think Ponium Selvan 1? No, PS1. Suddenly you will get confused. Correct. So within 30 seconds, if you have to find out the answer, then this is a logic you can use. 7 is odd number, 1 is odd number, 2 is even number, 6 and 8 are even number. So PS1 its reaction center is P700. That means it absorbs light of wavelength 700 nanometer. P680, it absorbs light of 680 nanometer. Okay, the reaction center. That's the way. So later, what all things are present in PS1 or what all things are present in LHC, light harvesting complex, in another session I will tell you. Okay. Now I want to concentrate just on to non-cyclic photophosphorylation. So sunlight energy fall on P700. Now P700, once you are absorbing light energy, you are getting excited, right? Additional energy you got. It enters into an excited state. So that's what I have represented by a star. Once it reaches an excited state, continuously it cannot absorb sunlight, right? It has to come back to the normal condition. For that, what it can do, it has to emit electrons. So it emits 2 electrons. These electrons are given for or taken by A0. What is A0? A0 is a modified chlorophyll molecule. Okay, modified chlorophyll molecule. Now it is taken by A1. What is A1? I'm writing it here, phyloquinone. Okay, phyloquinone. An electron carrier. Now from A1 it is taken by, so here we will write once again 2 electron. Okay, it is taken by FeXs. FeS, all these 3 FeS, XS, then SA, then Sp. All these 3 are iron sulfur containing proteins. Okay, so it is taken in this way. Now this electron is taken by ferredoxin. Okay, this is also containing iron. Okay, now what happens? It's also iron sulfur containing. Now from ferredoxin. So the electron is waiting here. Just imagine the electron is waiting here. Later I will tell you why. From here, the electron is waiting here. Now we will go to P700 again. So once the P700 will come back to its normal state, you have to imagine it lost electron, right? It has to get its electron back. The electron is already trapped here. It will not come back here. So that's why it is non-cyclic. Now what has to happen? So his buddy will come. P680 is going to help. Okay, actually according to the textbook, scientifically we have to study from here, but you won't get that logic. Okay, why P700 has to take the electron. That's why I'm starting from here, or I started from here. Now P680, light energy fall on P680. That means PS2, right? Now what happened? It enters into an excited state. Okay, now my son is also there. Sometimes I look at him. Now electron will go to where? Phosphorin. So here we are getting an important neat question. Which is the primary electron acceptor? Which accept electron from P680, the primary electron acceptor when it comes to non-cyclic photophosphorylation. Okay, non-cyclic photophosphorylation, it is always phosphorin, the primary electron acceptor, because I told you, you have to start the pathway from here only, the electron transport from here. So the primary electron transport, the primary electron acceptor in non-cyclic photophosphorylation is phosphorin. Okay, this is an important neat question. Never forget. Now from phosphorin, so I'm writing it here now, phosphorin. No need to by heart what is phosphorin, but you should understand, right? Phosphorin, you can say it's a modified chlorophyll molecule. Okay, now what is the difference between a chlorophyll molecule and phosphorin? Chlorophyll molecule, you know, right? In the center, right? It has a 4-pyrrole system. In the center, it possesses, right? That pyrrole system possesses in the center 1 magnesium. Now chlorophyll and hemoglobin, there is a difference because in the case of hemoglobin, the center ion is present. That is also an important neat question. Here you can see the center magnesium. Remember, chlorophyll, magnesium is present. Now, when the conditions are changing inside the cell, right? Sometimes what happens? Due to the changes in the H plus ion concentration, what is happening here? The chlorophyll molecule, it lost its magnesium. It loses its magnesium, and that time it's becoming phosphorin. Okay, that's the way you can say it's a degradation product of chlorophyll. Now from pheophytin, electron will go to QA. Okay, from there it will go to QB. From there it will go to cytochrome B6F complex. So this is called as cytochrome B6F complex. First I will tell you what is QA and QB. So here our electron reached which electron? The 2 electrons which is lost from P680. Now just imagine what are QA and QB. This we will call it as we will call them as plastoquinone. Okay, QA and QB are plastoquinone electron carriers. Okay, now cytochrome B6F complex. So I'm telling it is a complex. What is the speciality of complex? In this electron transport chain, in this non-cyclic photophosphorylation, the ATP molecules directly produced by utilizing the energy in this particular area when the electrons are moving in this cytochrome B6F complex. Here when it is moving, what is produced? The additional energy is given outside and by taking inorganic phosphate and ADP, ATP is produced, right? This you have to remember. Now cytochrome B6F complex means it possesses cytochrome B, cytochrome F, then there is a 17 KD protein or a peptide, and there is iron sulfur containing protein that is also present in this cytochrome B6F complex. That is not required for your need. Okay, now from cytochrome B6F, electron will go to PC. What is PC? PC is plastocyanin. Okay, it's plastocyanin. Now what is plastocyanin? Plastocyanin, just imagine cyanins. Okay, C is there. It's a copper containing protein. Okay, done. Now if you ask me what is pheophytin? Pheophytin gives electron from P680, or it is a primary electron acceptor of the non-cyclic photophosphorylation, right? Now it is the one which passes this electron to plastoquinone. Now if you ask me what is plastoquinone, it transfers electron in non-cyclic photophosphorylation between pheophytin and cytochrome B6F complex. Now what is plastocyanin? Once again you can create a definition. Plastocyanin, it's a copper containing protein. It's an electron carrier. It transfers the electron between cytochrome B6F complex and P700. Now wow, who is happy now? P700 is happy. Why? Because it got its electron. It already lost 2 electron, right? But it got its electron back, right? Now here what happens? Who lost electron? P680 lost its electron. Now he is also unhappy. He needs his electron back. So P680, once it lost its electron, it became a high oxidizing agent. Immediately, who is available there? Yeah, every cell has lots of water molecule, right? So it take one water molecule. We will call it as oxygen evolving complex. Now why it is called as YZ complex? Because this pathway is called as Z scheme, right? Because of the shape. So YZ complex or the oxygen evolving complex. Photolysis, water molecule. What will happen? Sunlight energy fall on it. Photolysis. The word we are using is photolysis. Break it. Lysis means breakdown. Photo means light. So the breakdown of water in the presence of sunlight is called as photolysis. It results in half O2, right? H2O, right? So half O2, 2H plus, right? And 2 electron. Now these 2 electrons will go to, yes, P680. So who is happy now? P680, he got his electron back. Now 2H plus, I told you, right? We were asking someone to wait here, right? Yes. Ferredoxin, NADP reductase. Already 2 electrons were there. These 2 electrons. Now what is happening? 2H plus. Everything is given to NADP. Now NADP is converted into NADPH plus H plus. So it results in the production of another energy molecule, NADPH2. Okay, nicotiniamide adenine dinucleotide phosphate. Okay, we are producing NADPH2. Now, who is poor here? Who lost everything? Who lost? That is another important need question. Who lost electron? Yes, of course, water lost its electron. Who lost hydrogen? Water, of course. Who lost oxygen? Water, right? So, this is non-cyclic photophosphorylation. Actually, we have to study from P680. But when you study from P680, I'm telling you 2 electrons are taken by P700. Suddenly you will wonder why P700 has to take 2 electrons. That's why I started from here, P700. So that you will understand P700 lost electron, it got its electron back. P680 lost its electron, it got its electron back. Now, poor water molecule, it lost everything. So to summarize, okay, in non-cyclic photophosphorylation, first point you have to remember, non-cyclic. The electron which is lost from the water, it is not coming back to water, right? Then, second point you have to remember, or the electron which is lost from P680, the same electron is not coming back here, right? When it comes to cyclic photophosphorylation, the same electron will come back, okay? Here, P680 is getting its electron from oxygen evolving complex, and water photolysis, complete breakdown happened. Second point, non-cyclic photophosphorylation possesses both the pigment system. PS1 is also there and PS1 is also there and PS2 are there. There is photolysis of water, there is evolution of oxygen, right? We will always tell in photosynthesis there is evolution of oxygen is occurring. So oxygen production is happening here. Then energy molecule, ATP is produced, yes. NADPH2 is also produced, yes. Right? So these are the major points you have to remember